The purpose of this project is to explore soil pH trends among the Platanus occidentales, known as the American sycamore, and the Pinus strobus, known as the eastern white pine. As opposed to deciduous trees, pine trees have been observed to tolerate more acidic soil. According to David South, pine trees growing in soil with a pH greater than 6 often exhibit chlorosis, or loss of pigment in their needles. We speculated that the pine needles themselves contribute to a higher soil acidity than the pine trees may need. As stated by Halil Sahlin, pine needles are composed of protocatechuic acid, which makes them slightly acidic. However, there may be another quality of pine needles that contribute to higher soil acidity. According to Sarah Moores, precipitation, specifically heavy rains, washes away alkaline or basic elements from the soil, leaving acidic elements such as hydrogen and aluminum. Unlike broadleaves, pine needles are light and thin, allowing more rain to seep into the ground and wash these alkaline elements out. With this information, we hypothesized that soil among the eastern white pine will be more acidic than soil among the American sycamore.
For each sycamore, we're measuring 1 meter out from the base of the tree. And then after we clear the leaves from the area, we're going to measure 5 centimeters into the ground to make sure we get our samples from the O layer of soil. And then we have a plastic bag for each sample and a teaspoon measurer. And we're going to collect 5 teaspoons of each sample, which translate to about 25 milliliters. And we'll get 5 scoops of that, and then close up our baggie and be done.
So for our experiment, we're pretty much looking at 3 different variables. One is the diameter of 10 sycamores. So this one would be 52 inches, which we will later convert into metric. The other 2 would be the canopy cover above, which would tell us how much sunlight our soil is getting for our sample. And the third one would be the percent of vegetation cover within 2 meters of the sycamore. So our slurry ratios is a 1:2, so it's 15 ml of soil and 30 ml of water. And our little scooper here is roughly 5 ml, so 3 scoops of soil per 1 little beaker of 30 ml. And then we stir that together, and that makes our little slushy, where it sits and waits for about 15 minutes so that the pH goes throughout.
Alright, so after we let our samples sit for 15 minutes, we're going to put them each on a stir plate, drop this little thing in so it can start to mix. That'll help us get a better pH reading, more accurate. And after that mixes for a little bit, we're going to go in with our clean pH meter, and we're going to look at the pH on here. We're using Logger Light to show the pH, so very gently, making sure the bottom doesn't touch the pH probe. And it looks like this one's about 5.9. So after that, we'll just take it out, turn it off for the next one. When we finished testing, we had 10 pH readings for each tree type. We compared these values by showing the average pH for each tree type. The average pH for the eastern white pine soil was 6.08, and the average pH for the American sycamore soil was higher at 6.45. Additionally, the error bars show no overlap, suggesting that the difference in pH may be significant. To further evaluate our pH readings, we performed a Mann-Whitney U-test to determine whether or not our values posed a significant difference between tree types. Since U and U prime were lower than the U critical value, we accepted our null hypothesis that there was no statistical significance between the values. Now, let's take a look at the relationship between pH and the 3 variables we measured. When comparing trunk diameter to the soil pH values of both trees, we saw little trend in either tree type. This data suggests that a larger trunk diameter does not enable either tree type to better influence soil acidity. When comparing canopy cover to the soil pH values of both trees, we saw a slight negative trend. However, the slope values near zero, suggesting that canopy cover generally has no effect on a tree's ability to influence soil pH. And finally, when comparing ground cover to the soil pH values of both trees, we saw a slight positive slope for the eastern white pine and a slight negative slope for the American sycamore. Again, both slope values near zero, suggesting that ground cover also has no effect on soil pH. In conclusion, the lack of statistical significance between the pH values lead us to reject our original hypothesis that soil among the eastern white pine is more acidic than soil among the American sycamore. Our research contributes to the idea that pine needles are not able to affect soil acidity. Likewise, many studies show that pine needles are acidic when they are fresh, but quickly lose the ability to influence soil pH as they dry out. Research that explores the effects of tree species on soil pH are important in understanding other ecological foci, such as niches in microbial communities.